molecular biology inevitably leads to molecular medicine. There's the history of medicine, and especially the history of medicine in the 20th century, is a subject in its own right, not exactly a science, but inseparable from science, not exactly an art, but inseparable from art in the classic sense of technique. And so we can't devote any attention to the history of medicine per se, but I think that in this lecture it would be valuable to see how the same intellectual influences that underlay the move to molecular biology also are at work in the shift of attention within medicine to what I have called molecular medicine. Medicine in the 20th century has itself become scientific in ways that it had not been prior to the 20th century, and reflects similar influences of physics and chemistry to the influences that were at work in research biology. For example, the work of physicists and chemists and the instruments that physicists and chemists have developed for their own purposes have been adapted to the uses in the medical community, as we will see in the last part of this lecture especially. But let's understand that medicine in the 20th century, building on a foundation of science, is associated, whether it's causal or not is another story, is associated with very dramatic changes in the lifestyle of the public in developed countries. For example, in 1900, less than 60% of women reached the age of 50 in the United States. In 1900, less than 60% of women in the United States reached the age of 50. In 2000, it's well over 95%. The life expectancy in the United States in 1900 was 47 years and a few months. Today, it's in the high 70s. And of course, probably around 80, closer to 80 for women, and maybe 77 or so for men. These are dramatic changes which are having profound demographic consequences for society as we enter the 21st century, and medicine is associated with this, and the competency of medicine is associated with this, although the extent to which sort of high-tech medicine can affect public statistics to that degree is a subject of controversy. So we have talked about molecular biology in very abstract and fundamental terms, but what is striking, and it's analogous, in my mind at least, it is analogous to the speed with which we went from the first satellites, which were almost toys, to increasingly competent spacecraft and satellites and research satellites and space probes. The same thing happened, an analogous thing happened in the wake of Watson and Crick's discovery of the structure of DNA. We are, I think, a bit complacent about the speed with which this very abstract fundamental work in molecular biology has begun to enter the mainstream of medical practice. It was 1957, just 4 years after Watson and Crick discovered the double helix structure of DNA, that Arthur Kornberg identified the enzyme responsible for sticking the bases together inside, the base pairs together, that are the genetic code. You know, the double helix is held together by base pairs that are the key to the protein manufacture. So Kornberg discovered the enzyme that will allow you to loosen those bonds. And in 1972, Paul Berg discovered or invented the basic techniques of recombinant DNA, of how you could disassemble and reassemble DNA molecules. You could cut them using Kornberg's enzyme, and then you could put them together again in new ways. Now, Berg was, between 1972 and 1975, Berg became increasingly concerned about the dangers that this represented for perhaps releasing new kinds of organisms, bacteria and viruses with modified DNA that existing organisms did not have defenses against. And so he rallied the biological research community in the United States, and in a very seminal moment, managed to get them to a conference at Asilomar on the California coast to talk about the ethics of recombinant DNA technology. This was in 1975. Berg himself unilaterally announced that he was stopping all research in this area for at least the next year. And he did impose a moratorium on himself, and he urged his fellow biologists to accept this moratorium on themselves until they could figure out safety procedures for working with recombinant DNA. As it happens, and perhaps not surprisingly, the conference ended with a view that this was dangerous, but we can control it. And a committee was formed, which together with the federal government worked out safety procedures for laboratories that do different levels of recombinant DNA research. But this is in the early and mid 1970s. By 1977, just 5 years after Berg invented the most primitive technique for disassembling DNA, the protein somatostatin, a hormone which is associated with inhibiting the action of the human release of the human growth hormone, was produced using this recombinant DNA technology. And in 1980, interferon, which is a protein produced by white blood cells as part of the immune system's response to antigens that enter into the blood, was produced by inserting a human gene, into a bacterium. Just a few years later, the FDA approved the manufacture of insulin, which until then, for decades, had been made by processing pig pancreases, an extremely a tedious process and a very messy one, by inserting a human gene for producing insulin into a bacterium. And this was commercialized so that insulin now all of a sudden was available, produced using recombinant DNA by bacteria, into which the human gene had been inserted. That was in 1980. It was also in 1980 that the first transgenic plants and animals were created, and we began to think that there was an industry waiting to be born here if we could transfer genes across plants and across animals. Then we could perhaps produce plants that had more attractive commercial characteristics and animals that had more attractive commercial characteristics in terms of cows, for example, and the milk that they produce, etc. So already by the 1980s, we have medical products of significance that are being produced by this technology. In the mid-1980s, the first monoclonal antibodies were produced. Monoclonal antibodies are exact clones of antibodies produced in response to a particular antigen or disease entity in the body. So that we can take a single antibody and clone it so that it is identical, and in principle at least, we can provide a person with unlimited exact copies of precisely the antibody that their own body produces in response to a particular gene. And if their body doesn't produce enough of it, why, we can now manufacture it in unlimited quantities and give them all that they need in order to respond to a particular disease. Now, in the 1980s and early 1990s, the hype over monoclonal antibodies far exceeded what was able to be done, but in the second half of the 1990s, monoclonal antibodies came back and are now a therapeutic tool that is available to physicians. In 1985, the human growth factor hormone was produced using recombinant DNA technology. And in 1986, for the first time, scientists were able to use recombinant DNA technology to combine an enzyme and an antibody, which opened up a whole new family of pharmaceutical substances for treating diseases, because now we could target cells that recognize the enzyme, and the antibody was carried along with the enzyme. The enzyme became a carrier for the antibody. And in 1997, the first artificial chromosome was produced, which again suggests in the 21st century that the potential power of molecular biology for providing increasingly powerful medical therapeutic tools. Now, not only at the level of the sort of the physical body, but there is such a thing at the end of the 20th century as molecular psychiatry. That is to say, looking at, and I referred to this in the previous lecture, reducing mental processes to molecular structural processes, the structure of molecular processes. And for example, there is apparently a strong correlation between suicide and the gene for the production of serotonin, which is a neurotransmitter substance in the brain. Similarly, there is a gene that's responsible for transporting serotonin, different from producing it, just for moving serotonin from one part of the brain to another, because it has to move around in order to carry the chemical message that it carries in the brain. That gene is associated with depression, and the two seem to go together, right, if you're sufficiently depressed. So the gene for serotonin transport, the gene for serotonin production. So here we have profoundly important human behaviors. That is to say, for the individual who suffers from depression, for the individual driven to suicide, these are human behaviors, and yet what we're saying is, I don't see, but let me put it differently, I don't see depression. What I see is a malfunctioning gene for the serotonin transport that happens to manifest itself in the big creature as depression, but it reduces to a structural problem having to do with the gene, with the way this gene codes for, it's not doing a proper job in providing for serotonin transport. And that may be analogous to Pauling's description of sickle cell anemia because of a structural problem. It's manifesting itself at the molecular level as a structural problem. And similarly, proteins have been identified that seem to be associated with schizophrenia. The gene that produces a protein called neuregulin, it seems to be strongly associated. There's a growing body of evidence that it's associated with schizophrenia. So here we see anorexia, schizophrenia, depression, suicide. More and more attention is being focused on molecules and that means their structural features and these behaviors. Now, I want to do two things with the rest of this lecture. One, to give you an overview of a beautiful case study by Louis Galambos and J.E. Sewell that indicates the way that the pharmaceutical industry has moved from 1900 to 2000 in the direction of molecular medicine, and then to talk about how medical imaging in the 20th century has moved in the direction of molecular imaging. Let me start with this case study, which they built around a small Philadelphia-based pharmaceutical company that was started by H.K. Mulford just around 1900 in Philadelphia. And it was started up on the idea of building a small, high-quality, high-tech, for the time, a pharmaceutical company that specialized in bacterial vaccines. We're more familiar with the idea of viral vaccines, but at the turn of the 20th century, in the early 1900s, bacterial vaccines were the primary tool for dealing with bacterial infections. And Mulford invested quite cleverly, recognized that he needed to invest in knowledge in order to create products that would give him a competitive advantage. And so what he did was he hired faculty from medical school, especially the Pennsylvania Medical School and Pharmaceutical College, and later on the veterinary school. He hired faculty members to be research consultants to help in the development of bacterial vaccines, keeping them always up to date, providing complete what he called use kits, so that the customer got everything in the package that was necessary to actually use the vaccine with the patient, and very cleverly with accompanying literature that carefully provided all the information that was deemed necessary for the user in order both to use it and to explain to the patient exactly what was going on. And Mulford's thrived for several decades, focusing on increasingly competent bacterial vaccines and also some serum antitoxins, which were popular at the time. In the 1920s, the company sort of lost its edge, and Sharp and Dohme a company that sold so-called ethical drugs, that non-biological drugs, wanted to get into the biological market, so they bought Mulford's company. They bought Mulford's company in order to get his network of customers and to get his technology. And so the company became part of Sharp and Dohme. And the CEO of the company, a new CEO of the company, recognized that the initial success of Mulford was because of the focus on research. And so they invested, Sharp and Dohme invested in the 1930s, a great deal of money in research in the latest technology, which, starting in 1935, we've already talked about this in a previous lecture, Gerhard Domagk invented sulfa drugs. The sulfa, S-U-L-F-A, have nothing to do with the compound sulfur. Sulfa drugs were a response to the observation that certain synthetic dyes killed bacteria. And Domagk followed this observation up, identified the dyes that seemed to have the most potent antibacterial qualities without having any obvious side effects, and he got the Nobel Prize for that. Although he was not allowed to collect the Nobel Prize because at the time Hitler was upset with the Swedish Nobel Prize Academy because they were giving Nobel Prizes to people that Hitler deemed unworthy of the prize. So Domagk was not allowed to go to Sweden to collect the prize. After the war, he was able to go and collect his prize at a special ceremony. However, the rules of the Academy do not allow giving the cash award if you don't get the prize within a certain narrow time limit. You don't pick up your prize. So after the war, when he probably could have used the money, he did not get any money for it, but he did eventually get the prize. But he did stimulate the creation of a new pharmaceutical industry based on sulfa drugs, which were antibacterial. I'm going to be careful. I don't want to call them antibiotics because that was 5 years later that large-scale production of streptomycin and penicillin, which we identify as antibiotics. Of course, the sulfa drugs are also antibiotic in the sense that antibiotic means anti-life. So it's they're all antibacterials, but sulfa drugs are not what we ordinarily think of as antibiotics because they are based on chemicals and in particular based on synthetic dye chemistry. So Sharp and Dohme focused on sulfa drugs, and then by the end of the 1930s on the new field of antibiotics, mass-producing penicillin and streptomycin, and then doing research on blood products, developing a very original technology for freeze-drying blood, which preserved it. And then World War II happened, and that was a tremendous boom for the company because the Army needed blood supplies for, obviously, to treat wounded soldiers. And this technology, which subsequently became a consumer food processing technology, instant coffee being an example, right? Dehydration and freeze drying. That technology was developed by Sharp and Dohme during the war, and so between the blood research and the antibiotics manufacture and the sulfa drugs, this was the line of development that Sharp and Dohme followed. And then after the war, when they had this tremendous body of knowledge about antibiotic production, they were forced, so to speak, by market pressures and government pressure to move to viral research because of the polio scares after World War II, especially in the late 1940s and 1950s. Polio epidemics in the United States, polio is caused by a virus, made the government push pharmaceutical companies into finding a response to polio. So they moved into viral research. That made them attractive to Merck. And Merck was a pharmaceutical company that actually was a supplier of fine chemicals to companies like Sharp and Dohme. And Merck decided they needed to integrate and to become more active in the pharmaceuticals because it looked like there was a lot more money to be made in pharmaceuticals than just in being a supplier to the pharmaceuticals company. So at the time, this was described as a merger, but in fact, it was a takeover, as we see by the fact that the name Merck, Sharp and Dohme eventually became just Merck. And Merck, again, invested heavily in research in viral vaccines. And this was in the mid-1950s initially, became involved in the mass production of polio vaccine, continued to invest further in viral vaccines, moved dramatically in, made a big commitment to recombinant DNA technology in the 1970s and 80s, and in fact, interestingly, by the 1990s, was now again making bacterial vaccines, which made Mulford famous at the beginning of the century. Right? They had moved from viral vaccines in the 1970s and 80s. Now, in the 1990s, they're making bacterial vaccines again, but these are bacterial vaccines that, using recombinant DNA technology, piggyback on viruses to deliver them. Viruses, as we all know very well, are really effective in getting their way into cells. If you can combine a vaccine with an antibiotic, a virus, I'm sorry, if you can add an antibiotic to a virus that you have defanged using recombinant DNA technology, so it can get into the cell, but it no longer knows what to do when it gets inside, but the antibiotic is now delivered. So the virus is now what's called a delivery vector. It is a vector, it is a transmission mechanism, it is a medium for transmitting the antibiotic to the cell. And this sounds like science fiction, but it is in fact a product that Merck has created based on recombinant DNA technology. And it's just kind of interesting that bacterial vaccines, which had been superseded by antibiotics, the original bacterial vaccines that had a philosophy of prevention. Antibiotics are a response to illness. You take the antibiotic when you're sick. The bacterial vaccines was to prevent, just as you get a polio vaccine to prevent getting polio. We've lost track of that. After World War II, bacterial vaccines more or less went away. We're familiar with viral vaccines, but they've actually come back in the 80s and 90s because of a number of severe bacterial illnesses that have once again become public health problems, even in this country and certainly in others. So there's one example, just one, and it is, I believe, representative, one example of how the pharmaceutical industry has moved increasingly in the direction of molecular therapeutic products. Now I'd like to spend some time talking about the imaging branch of medicine, medical imaging, which obviously has to begin with X-rays. X-rays also discovered in 1896. Just 3 years later, 1899, Thomas Edison had created the fluoroscope, which is one implementation of X-ray technology, and was manufacturing and selling fluoroscopes. Between 1899 and 1910, at which time there was a substantial production of X-ray machines for doctors and dentists' office, one must wonder at the stamina of our ancestors, because these machines exposed people to staggering doses of radiation, which often had severe and fatal consequences for the technicians and the physicians and the dentists who were using them, the nurses and so on. But we don't hear about massive numbers of people suffering from it, although individuals certainly did, because the exposure time of these machines was in minutes, sometimes 10 minutes or more of exposure. But in 1912, happily for all of us, a GE MIT faculty member who was also a consultant to General Electric produced an X-ray tube that only required seconds of exposure. And that became sort of a standard for the next few decades, and improved versions of that, of course, brought the time down even further. World War I was a major influence in that the Army wanted portable X-ray equipment to use in Europe, and thousands of people were trained to be X-ray technicians. When they came back after the war, we now had essentially a pool of equipment and expertise that made X-rays available. Now, why would people want to use them in very large quantities? Well, the answer came because of a real or perceived tuberculosis scare in the 1920s. And chest X-rays became almost universal. But one must wonder if the chest X-rays becoming universal was a factor that the companies had the equipment and the people and they wanted to use them so that they could get money from it, or whether we really all needed chest X-rays. that I'm sure is a very complicated question with no simple answer, but certainly it again reveals the way that circumstances and technologies and ideas and value judgments are enmeshed in complicated ways. X-ray technology developed in ways that we're familiar with, and the thing about it that I want to call attention to is that it's an outside-in imaging. Right? We bombard the body with X-rays. The X-rays pass through the body. They're absorbed by dense material like bone, and they go through the soft material, so you can image dense material using X-rays. But let's sort of humor me for the moment and accept that this is an outside-in kind of imaging. A significant improvement on X-rays was what we're familiar as CAT scans. Computer axial tomography scanning, which is the idea for which begins in the 1920s with a Frenchman who came to the United States and had this idea of rotating the X-ray machine around the patient to get a three-dimensional picture of what of the patient's body. And between 1929 and the 1970s, this was tried out in various places in Europe and the United States. And there was such a thing as whole body röntgenography, which is whole body X-rays in the 1930s. There were several companies that made prototype machines, but it was only in the 1970s that a commercial machine was invented in England. GE, maybe the best sense of the term, stole the market away from EMI, which was the British company that introduced the first commercial machine. They improved it, and they subsequently came to dominate the market. But CAT scans are interesting, I think, primarily because the underlying technology is based on a mathematical tool that has nothing to do with imaging, with medical imaging. The tool is tomography, which is a way of building a two-dimensional image out of one-dimensional data, a three-dimensional image out of two-dimensional data. Many scientists in many different disciplines use tomographic techniques, which are mathematical technique, for figuring out from a data set a more complex picture of what the data is describing. And it was recognizing in the 1970s, oh, we can adapt this technique to medical imaging that led to CAT scans. But that's still an outside-in technology. What has happened in the last third of the 20th century is that we have an inside-out imaging technology which is based on the activity of molecules inside the body. And these allow us to image not just things in the body, tissues and bones, but processes going on in the body. And that I think is that has given physicians and scientists powerful new tools for studying life processes. One of these, of course, is the MRI machine, magnetic resonance imaging machine, which is based on a discovery by the physicist I.I. Rabi at Columbia University in 1939, in which he was able to measure what's called the magnetic moment of a nucleus, the way that the nucleus is magnetized. And this led his students to develop, over the next 15 years, machines that scientists, physicists, and chemists used to do research, and that industry discovered could be used for testing materials by exciting the atoms in specimen materials, by imposing a very strong magnetic field around them, and then seeing how these atoms responded, in particular the hydrogen atom. Since we're mostly made out of water, and water is H2O, anything that excites hydrogen atoms gives off signals that we can build a picture out of. And that's what MRI machines do. And when in the late 1980s, superconducting magnets were added to these machines to get much more powerful fields, we have much more powerful images of what's going on inside the body anywhere there is a hydrogen atom, which is effectively everywhere in the body. So we're exciting hydrogen molecules. The other technology that images us from the inside out is PET scans, positron emission tomography. Notice tomography, it's the same mathematical technique, but now what we're doing is we're injecting into the body radioactive substances that decay in a particular way, and we're capturing the signature of that decay and building up an image of the organs and of the activity of the organs in which those molecules are contained. So we have here another example of molecular medicine of imaging the body from the inside out rather than from the outside in. Another instance in which we see the pervasiveness of this mindset and of the spread of the same ideas that underlie molecular biology.